Wow DIY. Today I will show you how to extract the DNA from wheat germ. All living things, from one-celled organisms to human beings, have their own genetic code stored in their DNA. Think of DNA as the building blocks of life. Stored in the cell's center, DNA defines all of our fundamental and distinctive characteristics, like our hair and our eye color. DNA is composed of two chains that coil around one another in the form of a double helix. If you stretched the DNA in one of your cells all the way out, it would be nearly 2 meters long. Isn't that incredible? Follow along with me to learn how to break open the cells of this wheat germ and separate out its DNA. This is one of our more complicated DIY activities, and you may need some adult assistance. First, gather some supplies. You will need 1 gram of wheat germ. You can find this at the local grocery store. If you don't have a kitchen scale, this is the equivalent of about ½ teaspoon. A liquid measuring cup with metric units or a graduated cylinder is useful, but I'm also going to provide you with the measurements in teaspoons. Prepare 20 milliliters or 4 teaspoons of hot water, 1 milliliter or about a quarter teaspoon of dish soap, 10 milliliters or 2 teaspoons of ice cold rubbing alcohol. You will also need vials or test tubes. Soda bottle preforms will also work. You can find something suitable at a craft store, on Amazon, or at a flower shop. The vials must be able to hold at least 50 milliliters or 10 teaspoons of liquid for the extraction, but a smaller one will work for storage. Finally, grab 2 wooden skewers and an eyedropper or a pipette. Now we're ready to get started. Begin by placing your rubbing alcohol in the freezer to cool it. Then measure out 1 gram or ½ teaspoon of wheat germ and place it in one of the vials. You can use an empty cup or a glass to support your vial. Carefully pour in 20 milliliters or 4 teaspoons of hot water. The water should be hot, but it does not need to be boiling. Have an adult help you though, so that you don't burn yourself. Cap your vial and shake it vigorously for 3 minutes. Next, add the 1 milliliter or quarter teaspoon of dish soap. Swirl it carefully and then invert it very gently for 2 to 3 minutes. Do not shake it, as you want to minimize any bubble formation. The soap is breaking open the cell wall and releasing the DNA. Now slowly add the 10 milliliters or 2 teaspoons of ice cold rubbing alcohol using the dropper so that the alcohol layer stays floating on the top. One useful trick to accomplish this is to add the alcohol along the inner side of the vial, like this. Where the alcohol and the soapy mixture meet, you will see a white substance begin to form. This is the DNA. Use the skewers to gently lift it to the top. Pour some rubbing alcohol in the second vial and then wind the DNA around the skewer to collect it and place it in the second vial to store it. Isn't science amazing? You can also try this experiment with blended strawberries or leafy greens. Just don't forget to clean up when you're done. DNA extraction allows scientists to improve the identification and treatment of many diseases. To learn more about what they do, search key terms like Sanger sequencing, PCR sequencing, gel electrophoresis, and DNA function and replication. Also check out the Human Genome Project. Thanks for joining me. If you want to find more fun DIY activities, visit us at wpafbstem.com. I'll see you next time.